A pleasant day to all. Welcome to the third part of the Enzyme lecture series, and today we are going to talk about enzyme regulation of activities. Regulation of enzyme activity within a cell is a necessity for many reasons, both of which involve the concept of energy conservation. First, a cell that continually produces large amounts of an enzyme for which substrate concentration is always very low is wasting energy. The production of the enzyme needs to be turned off. Second, a product of an enzyme-catalyzed reaction that is present in plentiful amounts in a cell, meaning it's more than needed, is a waste of energy if the enzyme continues to catalyze the reaction that produces the product. The enzyme needs to be turned off again. Metabolite flow tends to be unidirectional. Despite the existence of short-term oscillations in metabolite concentrations and enzyme levels, living cells exist in a dynamic steady state in which the mean concentrations of metabolic intermediates remain relatively constant over time. While all chemical reactions are to some extent reversible, in living cells, the reaction products of one enzyme-catalyzed reaction serve as substrates for and are removed by other enzyme-catalyzed reactions. So under these circumstances, many nominally reversible reactions occur unidirectionally. There are two general mechanisms that affect enzyme activity, and one is control of the overall quantities of enzyme or concentration of substrates present. Two, alteration of the catalytic efficiency of the enzyme. In general, there are properties when you call an enzyme regulatory. So, regulatory enzymes, these are enzymes that are found at or near the initial steps in a pathway or part of a branch point or crossover point between pathways. And then these enzymes are used to conserve energy, biosynthetic reaction pathways, which will not be operational unless a particular metabolite is required at a given time. So again, usually these regulatory enzymes, these are usually the enzymes that are rate limiting or committed step in a pathway, meaning that after this step, a particular reaction pathway will go to completion. So one example, of course, you have been memorizing this, the rate-limiting enzyme found in glycolysis. So these are examples of your regulatory enzymes. There are 5 ways in which you regulate an enzyme, and these are shown in this slide, the mechanism of enzyme regulation, and we will go to them one by one. First is by regulation through allosteric enzymes. So many, but not all of the enzymes responsible for regulating cellular processes are allosteric enzymes, and they have the following characteristics for them to be qualified as allosteric enzymes. Number 1. All allosteric enzymes have quaternary structure, that is, they are composed of 2 or more protein chains. 2. All allosteric enzymes have 2 kinds of binding sites, those for substrate and those for regulators. 3. Active and regulatory binding sites are distinct from each other in both location and shape. Often, the regulatory site is on one protein chain and the active site is on another. And the last characteristic, binding of a molecule at the regulatory site causes changes in the overall 3-dimensional structure of the enzyme, including structural changes at the active site. Thus, an allosteric enzyme is an enzyme with 2 or more protein chains, your quaternary structure, and 2 kinds of binding site, your substrate and your regulator. So, substances that bind at regulatory sites of allosteric enzymes are called regulators. The binding of a positive regulator increases enzyme activity. The shape of the active site is changed such that it can more readily accept substrate. The binding of a negative regulator decreases enzyme activity. Changes to the active site are such that substrate is less readily accepted. So this figure will show you the contrasts of the different effects on an allosteric enzyme, that is of positive and negative regulators that are produced. So the second process wherein enzymes can be regulated is through feedback controls or through feedback inhibition. Feedback control is a process in which activation or inhibition of the first reaction in a reaction sequence is controlled by a product of the reaction sequence. So this occurs when an end product of a pathway accumulates as the metabolic demand for it declines. This end product in turn binds to the regulatory enzyme at the start of the pathway and decreases its activity. The greater the end product levels, the greater the inhibition of enzyme activity. So this can either affect the Km or the Vmax of the enzyme reaction. So this is through feedback loops, and this is an illustration of the feedback control mechanism. You have to consider a biochemical process within the cell that occurs in several steps. So each step is catalyzed by a different enzyme. Note that the numbers would entail the enzymes and then the letters would entail your substrate and your product. So the product of each step is the substrate for the next enzyme. So what will happen in this reaction series if the final product, that's your letter F, is a negative regulator of the first enzyme, so enzyme 1. At low concentrations of F, the reaction sequence proceeds rapidly. At higher concentrations of F, the activity of enzyme 1 becomes inhibited by feedback, and eventually the activity stops. At the stopping point, there is sufficient F present in the cell to meet its needs. Later, when the concentration of F decreases through use in other cell reactions, the activity of enzyme 1 increases and more F is produced. A third mechanism for regulating cellular enzyme activity is based on the production of enzymes in an inactive form. These inactive enzyme precursors are then turned on at the appropriate time. Such a mechanism for control is often encountered in the production of proteolytic enzymes. So what are proteolytic enzymes? These are enzymes that catalyzes the breaking of peptide bonds that maintain the primary structure of a protein. So because they would otherwise destroy the tissues that produce them, proteolytic enzymes are generated in an inactive form and then later, when they are needed, are converted to their active form. Most digestive and blood clotting enzymes are proteolytic enzymes. So the inactive forms of proteolytic enzymes are called zymogen. So a zymogen is the inactive precursor of a proteolytic enzyme. An alternative but less often used name for a zymogen is proenzyme. So activation of a zymogen requires an enzyme-controlled reaction that removes some part of the zymogen structure. Such modification changes the three-dimensional structure, your secondary and tertiary structure of the zymogen, which affects active site conformation. So this is an example. For example, the zymogen pepsinogen is converted to the active enzyme pepsin in the stomach, where it then functions as a digestive enzyme. So pepsin would then digest the tissues of the stomach wall if it were prematurely generated in the active form. So that's why you have to keep it when you don't need it as a zymogen or an inactive form. So pepsinogen activation involves removal of a peptide fragment from its structure. Another example is your chymotrypsinogen. So when you don't need chymotrypsin, it becomes hidden as chymotrypsinogen. And when you have to activate it, it will be in a chymotrypsin form. Your blood clotting. So clotting involves a series of zymogen activations. There are 7 clotting factors, which are serine proteases involved in clotting cascade reactions. And of course, your insulin, when you still don't need that, you hide it as proinsulin. So, of the fourth mechanism, that's regulation of enzyme activity by covalent modifications. So, this is called covalent modification, and it involves adding or removing a group from an enzyme through the forming or breaking of a covalent bond. So, this is slower than allosteric regulation, it's more reversible, and it requires one enzyme for activation and one enzyme for inactivation. Covalent modification is a process in which enzyme activity is altered by covalently modifying the structure of the enzyme through attachment of a chemical group to or removal of a chemical group from a particular amino acid within the enzyme's structure. The most commonly encountered type of covalent modification involves the process by which a phosphate group is added to or removed from an enzyme. The source of the added phosphate group is often an ATP molecule. The process of addition of the phosphate group to the enzyme is called phosphorylation, and the removal of the phosphate group from the enzyme is called dephosphorylation. This phosphorylation-dephosphorylation process is the off-on or on-off switch for the enzyme. For some enzymes, the active turned-on form is the phosphorylated version of the enzyme. However, for other enzymes, it is the dephosphorylated version that is active. This is another mechanism that's regulation of enzyme concentrations, and of course, this is understandable. The more enzyme concentration, the more faster the reaction. So you really have to regulate them also. So this is signified by their turnover number, which is defined as the overall synthesis and degradation of a particular enzyme. And your enzyme concentration, one way of regulating the quantity of an enzyme is through this. So, how are the ways or what are the ways for regulating an enzyme? The amount of an enzyme in a cell can be increased by 1, increasing its rate of synthesis, or 2, you decrease the rate of its degradation or its destruction, or you can do both. So the last part of regulating your enzyme is by enzyme substrate compartmentation. So this just means that segregation of metabolic processes into distinct subcellular locations like the cytosol or specialized organelles can be used to regulate enzymes. So the substrates associated with a given pathway can also be localized to the same organelle or cytosolic location, and this allows for more specialized regulation of cellular processes. So this is by compartmentation. So one compartment, one job. So this kind of regulatory enzymes, they just do their process in this part of the compartment. They don't cross over and do the job there on the other compartments. So this table will show you specific organelles and their corresponding enzymes or the processes that they have to perform. So these are really centered. Like for example, in the nucleus, you see DNA and RNA synthesis, but you won't see that being done in peroxisomes. So this is just what it means by compartmentation. Putting them in specific compartments is one way on how to regulate their work. So regulation of enzyme is all that's there, and it's quite an easy topic as compared to the other parts. If you have any more questions, this is also a shorter chapter in Harper's, so you can read on it. Leaving you with this emoji, NZ, and leaving you with a message that you have to wear your mask at all times and of course practice social distancing. Have a nice day, everyone.